Welcome back. In the last lecture, I described the general principles of how drugs interact with the body, especially cancer drugs, how they're given to patients, and where they come from. Now I'm turning to specific examples and the science behind how the drugs work. You will see that some of the drugs work on DNA and others work on processes in the cells. And you'll recognize a lot of these processes from the rest of the course. My discussion in this lecture is not meant to be comprehensive, but to show you some representative types of drugs used and the scientific principles behind them. So in this lecture, I'm going to describe cancer drugs that attack DNA, and I'll also describe cancer drugs that attack general processes in the cell. Then we'll look at how chemotherapy sometimes has side effects, and very often does, and cells become resistant to chemotherapy. And finally, we'll talk about the hopeful aspects of targeted therapy, specific for a cancer. Now let's look at a cancer cell population. The growth of a cancer cell population is composed of two parameters: births, that is, cell division, producing new cells, and deaths, right? Cells dying, either being killed by the drug or going into programmed cell death. We can think of drugs that stop cells from dividing. The technical term for this is cytostatic, but just leave it at stop cells from dividing. Or we can think of drugs that promote cells to go into programmed cell death, apoptosis. Those are called cytotoxic drugs. In general, when we're looking at cells, we can look at drugs in a general way in this way. If you look at the overall scheme of proteins and how they're made in the cell, and this should be hopefully clear from previous parts of the course, we have DNA here, and DNA is expressed as RNA. Remember, a copy of the DNA is made of a certain gene that's being expressed, and then that is expressed as proteins. So now we're looking at DNA of oncogenes or tumor suppressor genes, and it's being expressed as a messenger RNA, which makes proteins that can be harmful. I've shown here a number of cancer drugs we will be looking at in this lecture and where they work on this scheme. For example, here's 5-fluorouracil looking at DNA and how actually the building up of DNA. Etoposide acts in the structure of DNA. You'll see how it acts. Adriamycin we won't talk about in detail, but it acts in DNA unwinding. Vincristine and Taxol are looking at a cell division protein, actually a similar one to that I discussed in the previous lecture. It's largely empirical which drugs work on which tumors. So physicians have a lot of drugs that can stop cells from dividing, and they try different ones on different tumors. But as you'll see later in the lecture, sometimes it's quite rational as to what drugs are designed, and this is where targeted therapy comes into play. To understand chemotherapy directed at DNA, let's review a little bit of the basics of DNA. DNA is a double helical molecule, and it's composed of these four building blocks, which we will abbreviate A, G, T, and C. And in the DNA double helix, where there are two strands of DNA, C is opposite G, and A is opposite T. So that's it for DNA. Drugs that target DNA do two things. They either target the building blocks of DNA, making up DNA, or they target the DNA structure itself. Let's look first at the building blocks, how you make the building blocks of DNA, how we make A, T, G, and C. And to do that, I want you to step back, as I am doing now, and consider the three basic theories of life, how scientists look at life these days. One was the cell theory. Life is composed of cell. A second was evolution by natural selection, change through time. The third I want to deal with here, which is called mechanism, which says we can describe life in terms of chemical transformations, in terms of chemical reactions. And some of these chemical transformations convert substances in the diet. Now, what's in your diet? Water and vitamins and proteins and carbohydrates and fats. And they take these molecules and they break them down into their constituent building blocks. So to give you just one example, when you eat a protein that has a specific shape, if you're eating meat, for example, or pasta, the protein has a specific shape, and then the protein is broken down to its constituent building blocks called amino acids. And so you end up with this soup of amino acids in the digestive system, then they get into the blood. And then the amazing thing that happens in the body, and the amazing thing of life is that the body can convert these amino acids into all sorts of other things inside the cell and into proteins themselves for the body's purposes. So I can take muscle, that is a steak that I'm eating, which is a muscle protein, and convert that into a protein that makes DNA building blocks in the cell or is residing in the cell nucleus. Amazing. And you saw very early in the course this confusing metabolic map of those thousands of reactions. Now, in doing chemical transformations in the cell, there are two things to keep in mind. First, the transformations are catalyzed or hastened by proteins called enzymes. And enzymes essentially move things along. That's what they do. They have a three-dimensional structure that moves things along. And the three-dimensional structure is highly specific, so it'll bind a certain chemical, and then convert it, and then it moves along to the next chemical change. Second, the second type of molecules involved in helping this are vitamins, and you'll see how one of them works in a moment. Let's focus on one conversion. Okay? So here is the conversion of DNA building blocks. And we start off with foods. They're digested in the body to the chemical building blocks of foods. And then these phenomenally amazing chemical changes happen, and we make A, T, G, and C. So we're zeroing in now on a certain type of conversions that are happening inside a cell. Normal cells as well as tumor cells. And we're going to look at making T, one of the building blocks of DNA. Okay, we'll have its name, thymine. Start with food. And the food breaks down into a number of molecules, and I'm going to show one of them here. That's one of the molecules in the cell that breaks down to. Don't worry, it gets converted. Many chemical reactions are making this molecule in the cell. And this molecule in the cell can be converted into T. That's the final product. That's T. Okay? Now, I told you that the conversion of molecules in the cell involves two things: an enzyme, a protein that hastens the reaction, and very often a vitamin or a helper that helps with the chemistry. Well, now we're going to zero in on the vitamin. And the vitamin is a derivative of folic acid that you're probably familiar with. And the folic acid does something. This folic acid in the body is converted to a molecule called tetrahydrofolate. Tetrahydro, 4 waters, folate. And the folate carries a group called CH3. And the technical term for that is a methyl group. So it's a CH3 group. And in the conversion of this molecule of the cell to T in DNA, only one chemical change happens. You don't need to be a Nobel Prize winner here to look at the difference between this substance and this substance. The difference is adding a CH3 group. So the vitamin carries the CH3 group and hands it off to that precursor molecule, and now you got T. Great. How are we going to manipulate this system to stop T from being made? Because if we do that, if we block one of the building blocks of DNA from being made, DNA will not be made, the cell can't divide, it just sits there. Great, and it'll probably go into programmed cell death. The way we do it is through a drug called methotrexate. Here is the same reaction, food and folic acid conversion into tetrahydrofolate is blocked right here. The drug methotrexate, here is a vial of it, stops that conversion. If you stop that conversion, there's no methyl group there. There's no CH3 group because they don't have tetrahydrofolate. The folic acid just sits there, doesn't grab this group. And so when the reaction tries to happen, it can't. You can't make T, and it dies what biochemists call a thymineless death. It's a horrible death. The cell dies because it can't make one of the building blocks of DNA. This is very useful in treating breast cancer, skin cancer, neck cancer, lung cancer. A side issue. How does this stuff really work, this methotrexate? I mean, I got the bottle, there's a stop sign there, right? Turns out methotrexate was a molecule that was made in the laboratory to look like folic acid. It's very cleverly made. It looks just like this vitamin. And so there's an enzyme that converts that vitamin, the folic acid, into tetrahydrofolate. The enzyme gets fooled. it doesn't bind folic acid, it binds methotrexate instead and kind of gets frozen in place. This is a really beautiful example of drug design. A chemist mimics a natural product, but does it in such a way that the whole reaction freezes. Problem with this drug is folic acid is used, tetrahydrofolate, in a number of other important chemical conversions besides making DNA, the building block of DNA. So there's going to be a lot of side effects. So let's try and find a drug that will be able to treat cancer cells and stop them from dividing, making T, by blocking this in another way. Well, there's another way to do it. The molecule here in the cell is converted into T, right? There's an enzyme here. An enzyme essentially is a protein there that binds the molecule in the cell. So if we could fool that enzyme, we might be able to stop the reaction. So let's design a molecule that looks like this, only fools the enzyme. We'll add something to the molecule. Here it is. The molecule is called 5-fluorouracil. And 5-fluorouracil has an F or fluorine atom right at an important point where it binds normally to the enzyme. And because it has that fluorine atom, the reaction doesn't happen, you don't get thymine. It's terrific. 5-fluorouracil is a widely used drug in cancer chemotherapy. Problem with it is it has to be infused over a long period of time. Patients very often have to wear a pump for a day or two at a time, and while it kind of seeps in, it doesn't dissolve really well in the fluid of the blood. And so instead, scientists have designed a drug called capecitabine that essentially makes 5-fluorouracil in a very short, in a short reaction, and it can be taken as a pill. So you take this as a pill, it gets into the blood, and it slowly leaks out 5-fluorouracil. So again, a terrific example of drug design. So I've talked about drugs that essentially block the building blocks of DNA. So for example, here's 5-fluorouracil, making up the building blocks so DNA can't be made. Now let me talk about some drugs that essentially bind directly to DNA and alter its function. In order for DNA to function, those two strands of DNA have to separate. Okay, so remember that. So they have to be available. They have to be available. Remember, DNA, 2 meters of DNA, 6 feet of DNA in a cell this big. It's huge. Each DNA molecule is a couple of inches long. In a cell, it's hundreds of thousands of a meter in diameter. It's so small. Teeny tiny cell nucleus. So the DNA is really, really a mess. Here's my metaphor for the mess. It's a big plate of pasta. So to make the DNA available, a region of DNA available for duplication of DNA or expression, we got to dig way into that pasta and get a small piece and get it up to the surface. There's a photograph right beside it. That's a beautiful photograph. It's a single molecule of DNA from a bacterium. A single molecule that's packed into that bacterium. It shows you the degree of packing that we have. By the way, a typical human DNA will be probably about 20 times longer than this. This is a single strand, a single DNA strand. It's really elegant to look at. And again, hard to imagine how it's going to unwind inside the cell because you're going to have to get it available first. How do you unknot something inside the cell? Well, really the only way in the end to do it, if we were doing it even with the pasta, if we had some glue to glue it together, is to have one strand pass through the other, to kind of undo it. The way we do that, it's an enzyme that does that. The enzyme has a name, it's called topoisomerase, but the name is not as important as how it works. Here's what we have to do. What we got to do is break this DNA. So there's the enzyme, which is this blue object. We got to break the DNA, which is the railroad track there, and then allow another strand to pass through. We're kind of unknotting the DNA. And there's a series of reactions that happens. Here's the other strand binding, and then it breaks the pink strand. You can see the strand now passing through, so it's now passed through completely. The DNA is now open, of course. You've got a gap in it because you broke it, and now you can seal it up. So the same enzyme seals it up. So this is an absolutely essential mechanism in the cell for making this mess of DNA a little bit available for duplicating, so it'll unwind, or being expressed. There's a drug. It's called etoposide. Etoposide comes as a natural product from the mayapple plant. It's been used as a medicinal by Native Americans for well, at least hundreds of years. The natural product was slightly modified. It's been used therapeutically as well as a drug. And what it does is it freezes this mechanism. It stops it right here. And so when DNA tries to be unknotted, right, it can't do it, and ultimately goes into programmed cell death. A terrific mechanism. A couple of more drugs that bind to DNA, just so you'll know their names and how they work. Cisplatin, very widely used drug, essentially is a drug that binds to DNA and works in a similar way to the story in the previous lecture of mustine. Remember the drug that came from poison gas? It cross-links DNA. So here's DNA replication, for example, where the strands of DNA have to separate. Here are the strands separating. The blue strands have to separate. They have to unwind. Okay? And to unwind the strands, you need to have them available to unwind. But if they're glued together, tough luck, huh? And so cisplatin does that. Essentially glues them together. Again, the cell go into programmed cell death because it can't duplicate or express its DNA. A couple of other drugs I won't show illustration-wise, but you'll see them in cancer therapy. A drug called cyclophosphamide adds chemical groups to DNA. Remember carcinogen in cigarette smoke and aflatoxin? They add these large groups to G on DNA, and then you get all these errors and mutations. Same thing with cyclophosphamide. It adds chemical groups to DNA, and you end up with mutations, and the cell becomes so mutated, it dies, programmed cell death. A drug called doxorubicin is shaped like a poker chip, and in the double helix, it wedges into the helix and stretches the bases, stretches the DNA building blocks, so that when replication happens, it makes more errors. Another drug called aniparib blocks the DNA repair system, so there'll be more errors as well. So the theme of these drugs is programmed cell death is going to happen if you damage DNA, right, of this particular class of drugs. Now, some drugs attack cell processes, so let's deal with some of those. One of the cell processes that's important is cell division. Cell division, as I indicated in the previous lecture, involves these railroad tracks called the cytoskeleton, called microtubules. So these railroad tracks are laid out, and you can envision the railroad car being a chromosome that moves along the railroad tracks. Taxol, you'll recall, bundles the tracks so they're not available. There's a whole series of other drugs that essentially stop these from being built. So the microtubule, this long tube, is made up of, in turn, by protein building blocks that go one on one, and you can see the building blocks there. And the chromosomes shown here move along it. So what vincristine does, and there are a number of other drugs that do, this comes from the periwinkle plant. It's a natural product. And it was used for a long time, over a century, in therapy, not chemotherapy for cancer, but for other purposes. Drugs like this were used, but we had no idea how they work. And this particular drug stops the microtubules from being made. And if it stops them from being made, no railroad track, no chromosome movement, no cell division. Another type of drug for cell processes are ones that target hormones. Many cancers are stimulated by hormones. Earlier on in the course, you remember estrogen replacement therapy and endometrial and breast cancer, for example. So estrogen acts by a hormone binding to a receptor protein, a system that I've described a number of times during the course. The receptor protein is highly specific for the hormone. And then the hormone acts, and then the receptor protein goes into the nucleus and alters gene expression, and so you get cell division happening. If we could block this interaction between the hormone and the receptor, and we put something on the receptor that kind of is a lock that doesn't turn the key, then you take the key, put it in the lock, break it off. That is a molecule that will fit into the receptor, but it does not cause the receptor to be activated. Then the estrogen can't bind, right? The molecule that I'm talking about is tamoxifen, which blocks the estrogen receptor. Now, there's another way to look at this whole system. If a hormone is stimulating cells to divide, And for example, prostate cancer cells are stimulated by testosterone. Estrogen stimulates breast cancer cells. Let's stop the hormone from being made. So there's a whole series of drugs for estrogen called aromatase inhibitors that are now active in cancer treatment that block the manufactures of hormones like estrogen and to other effects like testosterone. And you'll see them both in terms of cancer treatment and also in terms of prevention. So I've tried to give you a tour of some basic mechanisms of cancer drugs. I have to emphasize this tour is a brief one, and it's one where I've only given a couple of examples of certainly the most widely used drugs. But at least now you know some of the science behind how these things work. I mentioned that chemotherapy has to overcome side effects and resistance. If you know anything about cancer, you know about side effects. Side effects of cancer treatment are so bad in many cases that they limit patient tolerance of the drugs. And the patient just says, back off. Either I won't take any more or have to take less. And of course, if you take less of the drug, it might be less effective. What we're dealing with, of course, is the therapeutic index. The ideal therapeutic index says that the dose to kill the cancer cells will be much less than the dose that'll damage normal cells. And so we have a wide therapeutic index, and a wide therapeutic index means, great, no normal cell damage and no side effects. The reality is a lot of side effects happen. And you know from advertisements that you see if you're in America and you look at TV ads for drugs, 3/4 of the ad is looking at the side effects that can happen. In cancer, this is highly dramatic and very, very difficult. Some of these effects, we can figure out why they happen. For example, cell division is blocked. If you're taking a drug that blocks cell division, like binding to the railroad tracks, the microtubules, stopping them from working, or not being able to make DNA, then all cells that are dividing are going to be affected. So for example, the digestive system is constantly making new cells, stem cells. The bone marrow is constantly making new cells, blood cells. The skin is constantly making new cells because the cells slough off, and you've got stem cells there. Hair, right, constantly growing and being made by cells. So these side effects are rational. Another side effect, for example, is nervous system effects of some drugs that bind to microtubules, and that happens because in the nervous system, these railroad tracks are very important in the functioning. In some cases, it's not clear why the drugs cause effects. For example, nausea is a very common effect of cancer chemotherapy drugs. It's not exactly clear why different drugs cause nausea. Another effect that's really debilitating to patients is they become extremely fatigued. Why would it be that these drugs cause fatigue? Again, not that clear. Now, the side effects can be helped sometimes and sometimes not. This graphic shows you, for a number of the drugs we've dealt with, what the side effects are of some of the more common side effects. So, for example, bone marrow, digestive system, skin and hair, we've talked about already, right? And you notice here's some of the drugs: cyclophosphamide, methotrexate, fluorouracil, vinblastine, doxorubicin, all drugs I've just described. And you see that there's severe toxicity. The triple squares there indicate that can be dose limiting. In other words, the patient can say, I just can't deal with this. Doxorubicin damages the heart by mechanisms that are becoming clear but not terribly clear. Vinblastine or vincristine, this microtubule binding drugs for cell division, have nervous system effects. Some damage lungs, some damage kidney. So lots of things are happening. Some of these effects can be helped. One of the major advances in cancer treatment was the development of anti-nausea drugs in the late 20th century. So the cancer-fighting drugs were so causing of nausea that patients just couldn't stand it. And so now that it's somewhat alleviated by some terrific drugs that act on the brain. So nausea is alleviated. You can take bone marrow stimulants. There are drugs that will help with the bone marrow recovery. Sometimes bone marrow transplant will be useful, not always. There are drugs that will stop diarrhea, digestive system, kidney stimulants, heart medicines can improve things. So the idea of cancer chemotherapy is the cancer cells will recover from the dose a little bit, but the normal cells recover a lot from the dose. And so you do this fractionated dosing such that the cancer cells recover a little bit, but there's still a lot of damage there. The normal cells recover fully, and then you hit it again a couple of weeks later. So you got to be careful how much dose to give, lest you really make the patient sick. Drug resistance. In many cases, the initial treatment of cancer, it might even go into remission. You can't see it anymore, and then it comes back. So the cancer I study, which is small cell lung carcinoma, this is very common. The tumor shrinks to something clinically insignificant. You don't detect metastases, and then they're back. And when they're back, the tumor is resistant. So the drugs that were formerly okay at doses, reasonable doses, you can't use anymore. So you have to raise the dose of the drug, and if you raise the dose of the drug, you'll kill the patient. At that point, there's nothing you can do. So almost all patients with this type of lung cancer die with drug-resistant disease. Here is a very dramatic series of photographs of a patient with melanoma. So this is at diagnosis. You see the melanoma has spread. After 15 weeks of therapy, it's gone, but isn't. And it comes back after 22 weeks of therapy, and it's resistant at this point to the same drugs. Now the question, is resistance acquired, or is it there already? In this patient, were there tumor cells hanging around here already resistant at the very start of therapy? Or did those tumor cells develop as therapy proceeded and the tumor grew? It's probably a combination of both. The genome of tumors, as we know, is very prone to mutations and has poor DNA repair. So the tumor is quite heterogeneous and has a lot of DNA damage in it already. What are the mechanisms of resistance? Well, this is actually my field that I work on. It's a 10-alarm fire. The drug goes into the cell, and the cell has many mutations already that will allow it to be resistant. For example, decreased entry of drug into cells. I'll describe this in detail later on. Decreased activation of the drug, more inactivation of the drug, increased repair of the damage caused by the drug, alteration of target of the drug. There's a photograph there of the small cell lung cancer cells that I study. And you look at the cells, and they look just like small cell lung cancer cells. But deep inside that cell is an axis of evil, I call it. It's a series of mutations and of genes being turned on of all these systems that make it drug resistant. And the patient who has this type of tumor is not going to make it through therapy. Let me describe briefly some of the mechanisms of resistance. One of them is a pump, literally a pump at the membrane of the cell. So the drug in pharmacokinetics gets into the blood, and now in pharmacodynamics arrives at the cell, and the cell literally kicks it out. Great for the cell, bad for therapy. And so lots of studies are being done on this. This is one of the foci of my research and the research of many, many, many smart people trying to overcome this particular pump. A second mechanism is increased activation or inactivation of the drug. The liver system. You remember the liver system, and that actually exists inside cancer cells as well. Modifies drugs to make them active, so it might be turned down. Or it might be another system that makes the drugs inactive, so the drug gets inside the cell and then gets inactivated chemically, so it doesn't work anymore. Another mechanism of drug resistance is increased repair of cell damage. Many cancer cells have DNA repair, they're mutated, and they revert to normal, and they become resistant. Another example is alteration of the target. So the cell might have a mutation such that the target of the drug, the enzyme or the protein that's the target, might be mutated such that it doesn't bind. For Gleevec, for example, this is going on with patients. Their tumor cells are developing mutations, so Gleevec doesn't bind. So now we're at second and third generation of the Gleevec drug. So drug resistance is like a whack-a-mole at the fair, right? Every time you think you got it, another mechanism comes. Targeted therapy is relatively specific. So I've dealt with targeted therapy several times. So for example, Gleevec is terrific, right? It targets a certain protein that's made because of the shuffling of genes, the fusion protein. Let me just take you through one tumor to show you how targeted therapy can be valuable, and that's kidney cancer. Kidney cancer is about 60,000 new cases a year, 13,000 deaths in the United States. And a lot of kidney cancer is diagnosed by incidentally. Just you do a CT scan, you don't see any symptoms because it's only in one kidney, it hasn't spread yet, and then there it is. Previous treatment of kidney cancer, high-dose stimulants of the immune system, and it was bad. They were trying to get the immune system to fight the tumor. It wasn't working well. It was very, very difficult when tumor has spread. When they looked at the kidney cancer, they found two characteristics. Number one, it has extensive angiogenesis, a big blood supply, so maybe we'll stop that, try to target. And secondly, it has a lot of cell signaling going on. So a lot of cell signaling is turned on inside the cell, that multiple signals that turn on. So let's get drugs that'll target that. So they treat angiogenesis using, here's angiogenesis. Here's the signal from the tumor to the blood cell. And we're going to block it by angiogenesis. And here's a drug that does that. The drug is called sunitinib. It was designed for another purpose, but it turns out to block angiogenesis. Terrific, right? It's going to block the kidney cancer from getting angiogenesis. A second characteristic I mentioned is a lot of cell signaling. So here's a cell growth factor and a lot of signaling going on inside the cell. And it turns out the same drug, sunitinib, that blocks angiogenesis also blocks signaling. In fact, it was designed to block signaling and turns out to also block angiogenesis. So here's a single drug that's blocking the two things that make this tumor go. And here's the result, the initial clinical study. The y-axis shows progression-free survival, and the x-axis is time. But note, it's months, not years. Both graphs are going down. The graph of treating with the conventional therapy and the graph with the new therapy. So it's a clinical trial. So the patients are dying, but they're living for many months longer on the average. And some of them are living years longer. So there are many targets that are available now to treat cancer. Here are some of them, steroids and Herceptin, etc., and proteins expressed on breast cancer, etc. But here's the problem. Which tumors have which target? That's the difficult thing. We can think of two types of tumors, smart cancers and stupid cancers. Chronic myelogenous leukemia, the very first example I gave you in the course, is a stupid cancer. It's got a single important mutation, that shuffling gene, that fusion protein. It's got very few other mutations. You can take a single agent and knock it out, and it doesn't have resistance. Unfortunately, most tumors are smart cancers. They've got multiple mutations, a huge load. And the more we do genome sequencing, the more mutations we find. So you will need multiple targeted therapies, and resistance is very common. So this is the challenge we have. I've described cancer being treated with drugs in a general way, and now today in a specific way. And I've taken you from drug treatment, just stopping cells, to targeted therapies. The next lecture will look at the frontiers of chemotherapy treatment. Thank you.